स्टूडेंट्स, आपके एमएससी फाइनल बॉटनी का जो पेपर फर्स्ट है, प्लांट फिजियोलॉजी एंड मेटाबॉलिज्म, इसमें हम लास्ट टाइम पैसिव ट्रांसपोर्टेशन एंड एक्टिव ट्रांसपोर्टेशन पढ़ रहे थे. So in passive and active transportation, we could understand that a differentiation between two kind of transport is the involvement of energy. In the passive transportation, there is no involvement of energy, as well as this is downhill transportation. That means from its higher as according to concentration gradient, that means from higher concentration to the lower concentration. But in the case of active transportation, the transportation always to be done in uphill manner. That means against the concentration gradient, that means from the lower concentration to the higher concentration. So in the active transportation, when the uh, transportation is to be done as against the concentration gradient, so there is need to involve the energy as a ATP. So ATP hydrolysis gives energy to transport such kind of the molecules which move against the concentration gradient. So in the last time, we could also understand about the primary active transportation and the secondary active transportation. So in the secondary active transportation, that is the carrier mediated co-transport, that is the secondary active transportation. That means there is the involvement of two uh, and there is involvement of there is a movement of two molecules simultaneously um, in the same time. So this kind of the co-transport may be termed as the secondary active transport. Secondary active transport is driven indirectly by pumps. In plant cells, protons are extruded from the cytosol by electrogenic hydrogen ATPase operating in the plasma membrane and at the vacuolar membrane. The solutes are actively transported across a membrane against their gradient of electrochemical potential is by coupling the uphill transport of one solute to the downhill transport of another. So this is a coupling reaction. That means there is a one movement there are two molecules are involved in the transportation or the movement. There are there are two types of the secondary active transport, that may be symport or and antiport. In the screen you can see there is a three kind of transportation is shown here. In the first case, the uniport, the substrate is going to pass through the plasma membrane, in which you can see that only one molecule or the one substrate is passing from the outside to the inner side of the plasma membrane. In the second and third transportation, there is a co-transportation. In co-transportation, you can see symport and antiport process. In the symport process, two substrates are involved in the movement. S1 substrate and S2 substrate both are moving from from the outside of the cell to the inside of the cell through the plasma membrane in the same direction. But in the case of antiport co-transportation, you can see here in the screen that there is a involvement of movement. There is a movement of two substrates S1 and S2, but in the both but in the opposite direction. S1 is going to be moved from the outside of the cell to the inner side, while the S2 substrate is moving from the inside to the outside. But these both movements are, or the both transportations are simultaneous, and they are to be done in the same time. So antiport is the movement of two, or we can say the coupling movement in opposite direction, but in symport the coupling movement in the same direction. And the uniport, there is no co-transport, only one substrate is moving through the plasma membrane. So so now what are the here you can see another example. You can see the glucose is moving from glucose molecule is moving from outside to the inside. That is a uniport kind of the movement. Here you can see there is a sucrose and H positive. The both are moving in the same direction. The sucrose molecule and the proton generated from the proton pump is moving from the outside to the inner side in the coupling movement with sucrose. So this is sucrose as positive transportation, that is a co-transportation, is a kind of symport movement. Here you can also see the example of glucose, example of symport and antiport. The Na positive and the K positive movement is in the opposite direction. So ATP molecule is there to facilitate the movement of a sodium ion from the inside to the outside, and from the outside to inside movement of potassium ion. The same process, the antiport co-transportation. Here you can see in this example, calcium and Na positive sodium ions are moving to the sodium calcium exchanger through the plasma membrane. So these are the examples of the antiport. The sodium and glucose glucose molecule movements are shown here in the screen, and this is the symport kind of the active transportation. So basically there is a three type of transportation: uniport, symport and antiport. In the uniport, only one substrate molecule is involved there,